So as we continue looking at signal transduction, we looked at receptors and we looked at some generic second messenger signaling, the whole concept of this cascade. Well, I think you can understand too, when you look at receptors and their ligands, whether we're talking high affinity, low affinity, and when we say affinity, somebody just, can you explain to me what we're talking about with affinity?
The attraction.
That's a great word, attraction. How tightly they hold on to each other. Well, that's one way we can regulate the responses from the receptors and the ligands. We also saw with our dissociation constant, that effect, we're looking there at the concentration of a ligand to elicit a certain response. So if you have more ligand, you're going to have a greater response, right? Can you see how that will work? Depending on the affinity of the receptor and the ligand. Can you also appreciate that the number of receptors on the cell surface can affect a response?
Yeah.
So if you have more receptors, even if you have less ligand, you can still have a fairly robust response. Now the opposite is also true. If you have a lot of ligand in the cells, like, you know what, that's too loud. Like the car commercials when they come on television between the movie, you know what I'm talking about? And the volume goes through the roof. You turn down the volume. Cells can do that with their receptors in what is called receptor downregulation. Cells are constantly maintaining a certain level of protein receptors on the surface. As they get degraded, as they cease to function, they produce more and put them on the surface. Well, cells can also say, that's too loud, there's too much ligand, so they will basically endocytose nothing. And as you endocytose water, nothing, and you bring in that part of your membrane, guess what's coming in? Receptors. And they're brought in and they're destroyed because that was too loud of a signal for the amount of ligand in the space. So a lot of medications and a lot of drugs affect your cells in your bodies that way, because when you're exposed to daily amounts of a certain drug and you overwhelm the cells go, that's too loud, we're going to downregulate the receptor. So when you look at addicts, drug addicts, what do you know about that addict? The longer they're on that drug, what amount of drug are they going to need for the same effect? More and more and more, because the receptors are downregulated. So you can regulate responses from a ligand sense, concentration, and you can regulate receptors because you're just removing them off the surface because the signal was too loud. Now, we can also have something called desensitization. This gets into some molecular changes. You can add phosphates, and we're going to see some receptors do that to turn themselves on and off. But if you modify the cytoplasmic compartment of a particular receptor, you may change its affinity for the ligand. So instead of an on-off switch that we see with downregulation, it might be a volume switch. Okay, we're just not going to bind as tight, or we're going to bind a little more tightly. So we can do a lot with just a receptor, not taking into account the actual ligand. So as we mentioned our ligands and drugs, you're going to hear these two words quite a bit when we talk about pharmacologic agents. An agonist, this is going to be not necessarily a synthetic, but other than the naturally occurring ligand, this is going to be a different substance that can activate a receptor. And so while we have receptors in our body that binds to endorphins, which are natural painkillers, we can see that morphine will also bind to those same receptors, mimic endorphins, and you get pain relief. So that's an agonist, an activator. An antagonist. This is going to be a substance that inhibits the receptor, or we often call them blockers. So they can interfere with the ligand binding site of the receptor such that the naturally occurring ligand can't bind and can't activate. Agonists bind without activation. You see that caveat? Agonists bind and activate, antagonists block to prevent activation. And when we look at autonomic nervous system, for instance, in physiology, there's a whole category of drugs called beta blockers. Beta refers to adrenergic receptors. So when we say adrenergic, what substance are we really referring to when we use the term adrenergic? It's similar to cholinergic. You remember cholinergic? When we said cholinergic as a type of receptor, what neurotransmitter activates a cholinergic receptor? The first part of that word is the key, cholinergic.
It's not acetylcholine, is it?
It is acetylcholine. See, you wanted to go choline the first part. Actually, acetylcholine. All right. So cholinergic, acetylcholine. This is the one where you have to go kind of through the logic pathway. So an adrenergic receptor is going to bind which neurotransmitter? Oh, you're so close. Norepinephrine. Y'all were both so right there. Norepinephrine. Because I don't know. It was you had the lyrical part right, the number of syllables. Norepinephrine. Norepinephrine. So when we prevent the activation of these adrenergic receptors, which can lead to the desired physiological effect, in our case, the relaxation of smooth muscle. If someone is having an asthma attack, that means their airway are constricted. What do they usually take? An inhaler, albuterol. That's a beta blocker. It is a beta antagonist. Now that's probably the one you're more familiar with. You always see these folks with the inhalers. But you can also look at Tagamet, peptidase. These inhibit histamine binding to the receptors of your stomach cells that cause them then to produce the stomach acid. So we get the distinction between an agonist activator, an antagonist, a blocker, an inhibitor. So now we're going to check out two large categories of receptors as it relates to their ability to signal using second messengers. And we're going to start with our G protein-linked receptors, and then the next we're going to consider going to be the protein kinase-associated receptors. This is going to take probably the bulk of our time this morning. So G protein-linked receptors. These are going to be transmembrane proteins. What does transmembrane mean? When we talk about proteins? Through the membrane. This guy actually goes through 7 times. It's going to snake through up and down and up and down and up and down, and it's got 7 of these helices. Now what's another way that we can go through the membrane? Not helices, but the sheets, the beta barrels. It's still raining, I guess, huh? Man. So when we look at these very serpentine, and often these are called serpentine receptors. I think you can see like serpentine snake. These are going to have the ligand binding on the extracellular side, just like most of our transmembrane receptors. But on the cytoplasmic portion, we're going to see that this is where we have these loops that interact with this trimer of proteins called G proteins. And this is going to be the shuttle that then is going to relay the information that we had something bind to the receptor, therefore we need to do this. Now we also have a regulatory tail on the cytoplasmic side. that has these 3 amino acids that are very highly conserved. So this is significant to the cytoplasm of our G protein. So here we see sort of a ribbon diagram. There's our plasma membrane. We can see our 7-span protein structure. We have the ligand binding. It says messenger binding site on the outside. And here are our loops on the cytoplasmic face. Here's our GRK. This is a phosphorylation site, a regulatory domain. And then we're also going to have other sites in which we can bind with our G proteins. Now, in the little animation, we see our serpentine receptor, we see our ligand binding, and you can see how these 3 components of the G proteins go back and forth. You see that? This is going to be regulated by ligand on the surface, but it's also going to be regulated by, and in this case, this is our first time to see this, not ATP, but what? GTP. And the hydrolysis of GTP into GDP is an on-off switch for these G proteins. So do you see generically how this is happening? All right, so we're going to get into some detail about the molecular events of our G proteins. So we have our 3 subunits, alpha, beta, and gamma. And depending upon the cell and the receptor and what is being signaled, one or more of these will then travel through the cytoplasm and activate the next part of the signaling cascade. It may be the alpha subunit, it may be alpha beta, it may be the gamma. Now, together they're often referred to just collectively with all 3 of our subscripts. But understand, depending on what signals and what's downstream, it can either stimulate the cell to do something or it can inhibit the cell from doing something else. All we're doing is sending the message that says, ligand bound, you need to do something that you do because of ligand bound. Either you shut off or you turn on. So do you see the fidelity we can have here? It's not like in our nicotinic receptor. You remember nicotinic receptor? Where will you find a nicotinic receptor? What tissue in the body?
The brain. No.
And you're going to need this if you're going to get some work done. Skeletal muscle. The nicotinic receptors in skeletal muscle. Nicotinic receptor is going to bind what neurotransmitter? I will tip you off. Nicotinic is one of the 2 types of cholinergic receptors, acetylcholine. Now, when acetylcholine binds to the nicotinic receptor, that receptor is a closed ion channel. So when how many molecules of acetylcholine bind to open it? 2. Now, when you bind 2 molecules of acetylcholine, is there anything that channel is going to do other than open? No. It's just open or close. That's it. So that receptor, it can only do one thing. But with these G protein receptors, they go on and off, but the real business end is what's next. So do you see how you can now either turn on or turn off? It's not dependent on the G protein, but the next portion of the signaling cascade. Yeah.
So how do you know, is it always 2 that are required to make it fire, or it depends on that?
Now for the nicotinic receptor, it's 2. For our cases with most of our G protein receptors, it's just the 1. Nicotinic is the only one I can think of off the top of my head. I'm sure there are more, but it's the only one I can think of that will bind to activate. And if I can't think of it, then that probably means it wouldn't be on the test anyway, right? So here we have our ligand binding to the receptor. This is the activation cascade. The receptor either has been attached or it reattaches to our G protein complex, alpha, beta, gamma, all together. Now here's the part where our GDP comes in. When the alpha subunit is bound to a GDP. D is in David. That is the off signal for the G protein complex. So when the receptor and ligand get together, that's going to cause the alpha subunit to release that old GDP, and it's going to gain the GTP, which for the G proteins, that's the real on switch. That charges it up with energy. And when the alpha subunit binds to the GTP, then the whole complex, alpha, beta, gamma, separates from the receptor. Now you have a shuttle that can move and go to the next signaler in the cascade to then transmit the signal. So here in our diagram, here we have GDP on the alpha subunit. See how they're all three together? That's off. Nothing's going on. We don't have anything bound to our receptor. But when we bind ligand, it changes the shape of our cytoplasmic domain. The G proteins, they lose the GDP, pick up the GTP, bind to the cytoplasmic site, and that's what really facilitates this exchange of GDP for GTP, the binding of the ligand and the change of our cytoplasmic domain. And once we get that GTP in place, boom, it snaps apart. And for our example here, we're going to snap it apart into an alpha, and our beta gamma stay together. So we've got two units now. Instead of just one comprised of three parts, we have two units, one the alpha, and then the other one is comprised of the beta gamma still held together. And in a lot of cases, that alpha with the GTP is going to be the one that's going to run down the street and tell the next person, okay, you do this. Now that's the on switch. Once our alpha subunit gets to the next messenger in the cascade, that binding is going to lead to the GTP converting to GDP. Turns it off. Now the alpha comes back and binds to the beta gamma, and we're done. Now if ligand is still bound to the receptor, you're going to continue that cycle. Now understand, we've got hundreds of copies of our G proteins moving back and forth in the membrane. So these things are getting together by those random collisions. And it's not just this goes and does it, then those three have to go back together to the same receptor. Okay, they're just moving back and forth. So here we have our activated alpha binding to the next just generic messenger in our cascade. That binding causes the hydrolysis of GTP into GDP. Boom, come back together, ready to do it again. So what's the important part? That's what you want to know, right? What's the important part about G proteins? Activated by the receptor binding to ligand. Then it transmits the signal, this whole second messenger idea, transmits that signal to the next component in the cascade. Then it goes back to do it again. I mean, that's in a nutshell how this works. But what's the extra component that we didn't list in that illustration, bless you? The GTP, the energy required there. You can almost think of GTP like gasoline. You're not going to drive your car without gas. Now we're going to throw in a different level. Because in this case, we are not going to stick with just a generic second messenger that the G proteins activate. In this particular example, our second messenger is going to be adenylate cyclase. Adenylate cyclase is a transmembrane enzyme that can take ATP and convert it into cyclic AMP, which is a potent second messenger signaling that can go on downstream to activate something else. ATP is used for energy. What else is ATP used for? I'm sorry, I didn't hear you. Calvin cycle. We're going to use some in glycolysis, Calvin cycle, but where else will every single cell use ATP for something other than energy? We hear ATP energy, but what else do we use ATP for? adenosine triphosphate. Where do you use adenosine? It's a nucleotide. We use it in our DNA. So to me, it's crazy that the same thing that stores your energy is part of our genetic code, but it's got multiple uses. So cyclic AMP, very important. In our illustration, just like we saw, we have our receptor. Ligand binds to the receptor. There's our GDP in the inactive fate. We load it up with GTP. The alpha subunit slides over and activates adenylate cyclase to start converting ATP into cyclic AMP. And then that message is sent. So our alpha subunit signals adenylate cyclase. Adenylate cyclase says, hey, cyclic AMP, run down the street and tell this neighbor that we had ligand bind to the receptor. So do you see, we're adding and adding and adding. But as we add steps, what's one of the things that we're also doing? Amplifying the signal. And that's a big part of second messenger and signal transduction. You're amplifying the signal and spreading the signal through different pathways. So it's not just a straight line, one-shot thing. Here in our little animation, it's just, and I don't know why it stopped, but we can see when ligand binds, hopefully it'll do it more than that. We've got our G proteins and there's our adenylate cyclase. Now, whenever we bind ligand to the signal and our alpha subunit's going over to activate, we call that the alpha S. But there is also an inhibitory alpha that's swimming around in the system, and it can be used to inhibit the binding to adenylate cyclase. Now that's another regulatory step, right? You can block the binding, and adenylate cyclase says, okay, I don't care how much else you're sending, I'm not making any more. And this is just a naturally occurring negative feedback mechanism for preventing too much production of cyclic AMP, because that is such a potent messenger in many of our cellular cascades. So here's our adenosine triphosphate. Notice how with adenylate cyclase, we pull off the last two, right? Because cyclic AM, what does the M stand for? Mono. So the adenylate cyclase pulls off those last two phosphates, and you see how we're cycling the oxygens there of that phosphate group and making that little cyclical ring, and that's why it's called cyclic AMP, because of the ring structure. Now, you can degrade that, which is another means of regulation and turning it off, inactivating it, because we can take cyclic AMP with an enzyme called phosphodiesterase, remove the ring structure, now we just have AMP. This is not a messenger. Cyclic AMP is a messenger. It's kind of interesting that certain phosphodiesterase inhibitors can cause cyclic AMP to stick around longer and signal more. Guess what one of those inhibitors is? Caffeine. So that's, do you like caffeine? I say caffeine, she's like, oh, I love caffeine, especially on Monday morning. That's one of the mechanisms of caffeine and how it works. It keeps your body charged for longer. Now again, as we look at steps, and we're not going to have to remember these steps for any specific reason other than to show you we're amplifying the signal, diversifying the information. And at each step along the way, cyclic AMP, our next step, which is going to be the activation of a protein kinase, and we have a number of different protein kinases. Do we remember what a kinase does? The enzyme kinase. This might be one, this is one you just have to commit to memory, because proteases degrade proteins, lipases degrade lipids. This is a different one. A phosphorylase removes a phosphate group. Since we've already used phospho in a term, for the addition of a phosphate, somebody decided to name it a kinase. So again, you just got to commit that one to memory, because I don't know where they got phosphate and KIN, how they put those together. But we start cyclic AMP with our G proteins. Cyclic AMP then goes and activates protein kinase, in this case A. We're amplifying the signal, but at each step, it gives us an opportunity to further regulate the system. Because we can regulate G proteins, or we could regulate adenylate cyclase. Now we can regulate protein kinase A. What's a very easy way we can regulate those things? Have more or fewer numbers of the enzymes of the protein kinase. So do you see how the cell's got just a lot of fidelity in its ability to regulate the signaling that's happening inside of the cell? So once we turn on our protein kinase, that's going to add phosphate groups to a number of different proteins that is designed to affect. Now, cyclic AMP does more than just activating protein kinase A. It can activate quite a few second messengers depending on the cell and depending upon the availability of downstream messengers. But when we look at some of the major physiological consequences of cyclic AMP, bronchodilation, the prevention of platelet aggregation, you know that's pretty important. Your body spends a lot of energy preventing blood clots because you don't want the blood to clot just in your circulation. If a blood were to clot and stick in your heart, what do you got? Clog up a blood vessel in your heart, what are you going to have? Heart attack, myocardial infarct. Clot up the blood, it sticks in a vessel in your brain, what do you got? Stroke. Yeah. Now in your heart, in your lungs, you can have an aneurysm or an embolism where you block the blood going to your lungs. Either one's a really bad day. So, yeah, we're going to prevent platelet aggregation, blood clotting. Cyclic AMP works to reduce inflammation. So, just to give you a little bit of context, we're not just learning this stuff so that some nerd in a lab can further investigate what it's doing. Now, we know the downstream consequences of what's going on, and I think you would agree that's pretty important for our day-to-day homeostasis and health and well-being. So adenylate cyclase activated by the alpha stimulatory subunit. Well, in some cases, the beta gamma is the signaler. Alpha's just kind of hanging out. And here we have our second type of acetylcholine receptor that is called a muscarinic type of receptor. Now, nicotinic. What tissue did we say nicotinic receptors were in? What muscle? Skeletal muscle. Commit this to memory. Write that down. Nicotinic acetylcholine receptors are found in skeletal muscle. You're going to see this when you get to physiology, so go ahead and learn it now. The muscarinic type of acetylcholine receptor is found in the other types of muscle. What are our two other types of muscle? Smooth and cardiac. So if you remember acetylcholine receptors and muscle, nicotinic receptor in skeletal, muscarinic in cardiac and smooth, that's going to take you far when you get to autonomic nervous system in physiology. But our muscarinic receptor in the smooth and cardiac muscle, it binds to a single molecule of acetylcholine. It is a G protein-linked receptor. That's how they're often described. And you can see with the binding of acetylcholine, activation of the G proteins, the GTP added to the alpha subunit that release, that causes beta gamma to go over and activate an ion channel. And in this illustration, we have a potassium channel that's closed. Once those alpha betas come over and open the ion channel, potassium can diffuse. Potassium is usually in highest concentration where? In or out? Inside. Because our buddy sodium is high outside, and sodium and potassium are what? Opposite. Almost always opposite. Now, if you don't think G proteins are important, look at some of the problems that can happen when something messes it up. You ever heard of cholera? Not a good thing, right? Cholera toxin prevents the alpha subunit hydrolysis of GTP. What does that mean? If you can't hydrolyze the GTP on the alpha subunit, what can you not do to the G proteins? You can't turn them off. You can't turn them off. They're going to keep signaling and keep signaling and keep signaling, which is going to cause cyclic AMP to work overtime. And if all of this is happening in your cells of your intestine, that's going to cause your intestinal cells to secrete a ton of salt, and the water follows. Do I have to paint you a picture of what that's going to look like for that patient? It's going to be Montezuma's revenge times 100. Yeah, massive, massive diarrhea. And cholera is a deadly disease. Which just kind of brings you to a point. Do you realize how many thousands of people die every year from diarrhea? They die. Can you imagine dying from diarrhea? That would be the worst form of punishment that I could ever inflict on the worst criminal ever. Yeah, just death by diarrhea. But because they're drinking contaminated water day in, day out, and they have these parasitic infections and other diseases, they dehydrate because of the diarrhea. All it would take is just some clean water, and thousands of people could be saved. It just blows my mind. In our technological age, we still have people dying from diarrhea. Sorry, I didn't mean to get on that soapbox, but whooping cough toxin. Whooping cough is going to prevent our GI, the inhibitory G protein. That's going to prevent it from inhibiting. Well, it's the same thing. But in this case, instead of this happening in intestinal cells, it happens in your lungs. And if you've ever heard someone with a whooping cough, it is such a distinctive sound, you will never forget it. And that sound is because of the resonance that happens because of the fluid in the lungs. The coughing happens because you're trying to get the fluid out of the lungs. And it's all affecting G proteins. So just those two examples illustrate how important G proteins are. They're not the receptors. They're not even the ligands. They're just the part of the signaling cascade that leads to such horrible things.
So are we going to know specifically like whooping cough toxin prevents GI protein, or is it just going to be like what can kind of be affected by the G proteins? How specific is it going to be?
Well, let's just, okay. You're studying for the exam. So distill it down. Whooping cough knocks out GI, can't turn off adenylate cyclase, fluid in lungs. Cholera prevents GTP hydrolysis alpha subunit, can't turn off adenylate cyclase, too much water in the intestines. Because really the effect is too much water, either in the lungs or in the intestines. How you can, and it's the inability to turn off cyclic AMP. They're just two different ways that you can't turn it off. Whooping cough, the inhibition doesn't work. The GI doesn't work. And in cholera toxin, the GTP doesn't work. So do you see how you distill that down to these nuggets that you can hang on to? It's not the whole slide. It's not the whole illustration. But it's enough information that you can understand it and use that nugget to analyze whatever the question might be. And again, to me, learning just as much and doing less work. It sounds lazy, but if you have too much information, your brain's not going to remember any of it. Have you ever had to do something and there was so much that you just didn't even start? But if you can break it down into smaller chunks and smaller and look like it's doable, then you have a whole different feeling before you start something. That I like doing just that, making as smaller chunks as I can. And when you get to physiology, there's a lot more to learn in physiology than in here. And so I usually start my first day lecture in physiology with don't be overwhelmed yet. But physiology is like eating an elephant. How do you eat an elephant? One bite at a time. I just like having small bites. I don't look at the whole thing of what I have to do. As a student, I usually freaked out after the first day. When I got the syllabus, I freaked out because I got to do all this like I had to do it that night or something. But then you realize, okay, I've got to do this today, this day, this day. So make those small nuggets that you can hang on to. Now, not related to G proteins directly, but another component of our signaling cascade is going to be calcium. Calcium in your bones. Calcium is required, remember, in the end of our axon. What is calcium required for relative to our axon? Calcium, our voltage-gated calcium channels, as the action potential comes down, they open, calcium comes in, starts a second messenger signaling cascade that leads to the secretion of our neurotransmitters. So we've already seen calcium being used, so it makes sense that we would put calcium in here. Now, where's calcium at the highest concentration? Outside. So when you open those voltage-gated channels, that's why calcium comes in. Physiology, we're going to see calcium coming into the cell, the cytoplasm. That's going to lead to a muscle contraction. The way that you relax the muscle is get calcium back away from the inside of the cell.
Is that the same with the adrenaline cyclase, that it's making a stronger signal by adding the calcium?
The adenylate cyclase doesn't directly do anything with calcium. The cyclic AMP may be a consequence of calcium coming in, or the cyclic AMP may signal to allow calcium to come in. But adenylate cyclase doesn't have anything directly to do with the calcium.
But the calcium comes in to create a stronger signal?
Yes, but we're going to see how that works, I think in just a slide coming up after this. Because in our axon, calcium comes in and it binds to a calcium binding molecule that I think is on the next slide called calmodulin. That turns calmodulin on, that it can go turn on a protein kinase and da-da-da-da-da. All those dominoes start to fall when calcium comes in. All right. So when we look at, for instance, egg fertilization, calcium's going to be stored inside the egg, all right, in compartments. And when you have contact of the sperm with the egg membrane and they fuse together, that calcium is going to be secreted out into space. It's called the cortical reaction. That's going to cause that membrane to almost become toxic to sperm. And the sperm can't bind, they can't fuse, and can't penetrate, because I think you can understand if two sperm fertilize an egg, that would not be good. Too much genetic material is a horrible thing. You certainly do not want 3 copies of the genetic code.
Has that ever been done?
It's not compatible with life. And you can wrap your brain around this. Down syndrome. What causes Down syndrome? One extra chromosome of number 21. Imagine having an extra chromosome of every numbered chromosome. And in fact, most trisomies are lethal. Just had some friends celebrated, I guess not celebrated, commemorated, probably a better word, their stillborn baby. That was this past weekend. It had been a year since the baby was stillborn. Trisomy 13. Trisomy 13 is one that often they will get to the stage of delivery. Most trisomies are spontaneously aborted. It's just, that's how bad those are. So an entire genome, no. That's going to be a spontaneous abort, just not compatible with life. So that would be a critical physiological event, preventing polyspermy. Prevent the fertilization of more than one sperm. And calcium plays a huge role in that process. So there's our calmodulin. So in our axon terminus, calcium floods in because of the action potential coming to the end. Calcium binds to calmodulin, causes calmodulin then to go turn on the next partner in our signaling cascade. In most cases, it's a protein kinase. That's what's happening to allow the release of our neurotransmitters into the synaptic space. So in this case here we have acetylcholine binding to our receptor. We'll just go ahead and say that that is what type of acetylcholine receptor. Is that going to be nicotinic or is it going to be muscarinic? Muscarinic. Why did you say that? There's only one acetylcholine binding, but we also know that the muscarinic receptor has to do with G proteins. G proteins are in here somewhere. That's probably going to be from this number 1. Oh, wait, there it is, G protein link. So it's got to be muscarinic. They're just not showing the G proteins. But they do show calcium being released from the endoplasmic reticulum. In our muscle cells, we call it sarcoplasmic reticulum, but it's still the ER. Calcium binds to calmodulin. Calmodulin's then activating nitric oxide synthase. That then goes on, does some other things through our other signaling molecules. There's cyclic AMP even in this one. So in this case, the G proteins, cyclic AMP, calcium, calmodulin, it's all different steps in this whole cascade that leads to muscle relaxation. So with each step, do you see the complexity? But can you, again, appreciate how many steps there are places where you can regulate and the amplification that can happen? From one acetylcholine, if you're activating hundreds of calmodulins, you're activating thousands of guanyl cyclase, tens of thousands. Hopefully you can envision that amplification of our intracellular signals. Now our last group, protein kinase associated receptors. Protein kinases, what do they do? A kinase adds a phosphate group to proteins. And in many cases, this is going to be to a tyrosine amino acid within the primary sequence of our proteins. But you can also have some serine, threonine kinases. Simply, these are the amino acids that are going to have the phosphate added to them in those particular locations. There's a bunch of them. There's a bunch of these receptors, and they have a huge variety of cells that you find these types of receptors on that lead to a ton of different physiological consequences, not the least of which that leads to the secretion or exocytosis of materials from the cell that affects the local environment. Now, you don't have to memorize this list. This is just giving you some ideas. What do you see in common with all of these names? Well, most of the names. Epidermal, what's that? Growth factor. Transforming, growth factor. Platelet-derived, growth factor. So many of our growth factors are going to facilitate that second messenger signaling through receptor tyrosine kinases or protein kinases. Now, these are going to be different from our G proteins because those receptors had 7 spans, remember? These are just a single linear transmembrane molecule. Outside binds to the ligand or growth factor. The inside's got a cytoplasmic tail. And that cytoplasmic tail is where we're going to see this kinase activity. And for our tyrosine kinases, we're going to have a number of these tyrosine, where you see residues, it's amino acids, just in certain locations in the cytoplasmic domain. And once we bind the growth factor, these receptors are going to come and they're going to get very, very close together. And these cytoplasmic tails with these activities are going to phosphorylate their neighbor. Now, some of these can phosphorylate themselves, but mostly they're going to phosphorylate their neighbor. That then leads to the activation and the downstream signaling. Now, in some cases, you may see proteins that are referred to as non-receptor tyrosine kinases. These are simply other proteins that interact with the actual receptor that leads to the ability of that receptor to phosphorylate the tyrosine amino acids. So it can either be just one long protein, or it may have an associated portion, like a second protein, so our quaternary structure, that it takes both components to be able to fully activate that particular receptor. So I got ahead of myself with this slide. This talks about the phosphorylation, how these two receptors can phosphorylate each other, or they can autophosphorylate themselves. I was trying to get to this picture. So do you see our single tyrosine kinase receptor? Here we have, in this case, epidermal growth factor. There's the binding site on the extracellular domain. There's our transmembrane domain. We have the kinase activity, and then we have the tail that has all of the tyrosine residues. Binding of the growth factor and ligand causes them to get close together, and now the tails in each come up into the catalytic domain, and you see how these two on the right, how they're each phosphorylating the tyrosines of the neighbor. That's going to be the activation then that happens because we bound the growth factors to the surface. The phosphorylation of these tyrosines in the cytoplasmic tail is the signal to then go on and activate the second messenger and downstream and so on. But you see how this differs from G proteins? G proteins are these other things that have to be there. These tyrosine kinases all do it just themselves. Now again, epidermal growth factor as one of our examples. We've got a lot of other downstream signalers that are involved. But those details of the downstream signaling, basically let's look at the bottom line. When you go through all of these signals, and look, we've got a bunch of kinases here, MAP kinase, we've got some MEKs. The whole point of this, again, is the amplification and the ability to regulate signal. But really, the point is the growth factor binding outside is going to lead to a change in gene expression. Everything the cell does is related to gene expression. And most of our growth factors, I think you can envision, are going to cause changes in cell proliferation. Fibroblast growth factor. If fibroblast growth factor binds to the FGF receptor on a fibroblast, what do you think is going to happen to that fibroblast? It's going to grow faster. It's going to undergo cell division, make more fibroblasts. You see how that works? Now, this is also a bad situation because if you lose the ability to control your signal transduction at any of those steps, what might happen? So you got a lot of spots where you can regulate, but now you've also got a lot of targets for mutations. And mutations are what leads to then cancer. So it's a trade-off. By having such diversity, you open yourself up to the potential for problems. So we've talked about our G protein links. We've talked about our protein tyrosine kinases. Now let's get back to looking at the actual ligand, the signalers. And in this case, we've got hormones. Now, hormones, unlike some of your other, like neurotransmitters, for instance, like acetylcholine, norepinephrine, those are going to be secreted very locally, that 20 nanometer space. Hormones, they can be produced in the pituitary gland, which is at the base of your brain, and make their way all the way to your big toe. Because these are traveling via your bloodstream. And if these hormones are placed into your bloodstream, we call them endocrine hormones. If they're placed into a duct, if secretions are placed into a duct and transported to a specific place, we call them exocrine secretions. So we've talked about epinephrine. Epinephrine is a catecholamine. It's derived from tyrosine. And so it's an amino acid derivative. Insulin We know that's pretty important, right? To balance our sugar in our bloodstream. Insulin's a protein. Testosterone, one of our androgen hormones. Well, this is going to be made from our sterols. So you got to have cholesterol if you're going to produce some of these testosterones and your other estrogens, as well as the androgens. We can have short amino acid sequences, peptides, which is usually how that's different than our proteins. Peptides are shorter. And we have this hormone, anti-diuretic hormone, ADH. It's also known as vasopressin. In physiology, you're going to understand that that's going to really balance your body's ability and your kidney's ability to conserve water. The more ADH that's produced, the lower the volume of urine you secrete, the more water you retain. And vice versa. You shut off ADH, you're going to produce more urine. So a lot of regulation happening because of hormone production and certainly because of hormone receptors. Epinephrine. Sounds like its cousin. What's the neurotransmitter cousin of epinephrine? Norepinephrine. They bind to many of the same receptors. They lead to many of the same effects on your body. It's activating the fight or flight nervous system. I mean, this thing's called adrenaline. And it's really going to get your body ready to do something, meaning it's going to open up blood vessels going to your skeletal muscles. It's going to jack up your heart rate. It's going to open up the bronchioles of your airways. But while we're activating all these things, it's also going to cause the muscle of your GI tract to just relax. Because during a fight or flight response, your gastrointestinal tract shuts down. You're not going to digest anything. The peristalsis, it's moving material through your intestinal tract, stops completely. And the blood is actually shunted away from your GI tract. That's why if you eat a big meal in the summertime, what should you not do for like 30 minutes? Go swim. Has your mom ever said, oh, you can't go swimming, you got to wait 30 minutes? Like, what are you talking about? That's because you're going to have this big meal sitting on your stomach. Your stomach's not going to process it if you're out there being real active and swimming around, because that's a more active fight or flight autonomic response. And you could get cramps or you could get just indigestion. So here's epinephrine and norepinephrine. Yeah, we've got our adrenergic receptor, and we can see norepinephrine can bind, but epinephrine can as well. What do we have here? What's our cluster here in the cytoplasm? It's our G proteins. So many of our adrenergic receptors, see this is the alpha 1, here we have a beta adrenergic receptor. Many of them are G protein mediated. And there's our friend adenylate cyclase. So albuterol, that beta blocker, that's going to prevent the G proteins from coming over and activating adenylate cyclase. But in this case, for a different receptor in a different cell type, when you have the binding of epinephrine or norepinephrine, the G proteins, they don't go to adenylate cyclase. They come over to a second messenger, a different one. This is called phospholipase C, that works on some of our phospholipid signalers that then go on and begin to activate some of our protein kinases that then go on downstream. Oops, what's activated here? Calcium, calmodulin. That goes on to lead to contraction of our smooth muscle. So you see, same hormone, different receptors. Maybe we use some of the same second messenger signalings, but you can have very different effects. That again plays into the fidelity, the diversity of responses in our different tissues, and that's why we don't have to have a ton of different hormones to cause all these different things to happen in your body. We just need different signalers and sometimes different receptors to get the desired physiological response. Insulin, this is protein. Your blood sugar is high, insulin's produced, cells take up the insulin, store it as glycogen. Here's our insulin receptor. What category of receptor would you call the insulin receptor? This is our tyrosine kinase-like receptor. And you can see once you bind insulin, that's going to cause this activation of a kinase. The kinase is over here activating our PIP, second messenger signaling. You see we're adding a phosphate to that. That's going to activate our enzymes. It's going to cause glycogen to pop off individual glucose molecules that then can be transported into the cell. Now, this is a preview of chapter 19 only because it relates to our second messenger signaling. Are we all familiar with apoptosis, aka programmed cell death? There's so much signaling that goes on inside that cell that leads to the destruction of the cell and the shredding of our DNA. Namely, activation of these enzymes that are in the cytoplasm called caspases. And so when we look at the signaling involved, we have BCL2. These are somewhat of our three triggers for apoptosis. So make sure you know these three triggers. BCL2, when you see this on our mitochondrial surface, that means that mitochondria is healthy, happy, functional. When BCL2 gets downregulated and you have less of BCL2 on the surface, that's going to be a signal to start killing the cell because that mitochondria is getting bad. Cytochrome C, this is going to be an important component in our electron transport chain producing energy. Well, if you have a cytochrome C in the cytoplasm, meaning it got out of the mitochondria is bad, the mitochondria is dead, boop, that's going to lead to the activation of caspase. P53, this is going to be another component of our mitochondria, electron transport chain. When P53 is in the cytoplasm, you see the message here? If the mitochondria is bad or shutting down, that cell's not going to make it. So let's just kill that cell, get it out before something bad can happen. So BCL2 goes away, cytochrome C, P53 end up in the cytoplasm, boom, that cell's going to die. So there's our BCL2, which inhibits apoptosis. But if cytochrome C gets out here, there's our P53. All that is sort of further downstream of other effects that can happen, because did you know you could actually signal a cell to die? Cytotoxic T lymphocytes, if they detect that a cell is virally infected or is a tumorigenic cell, it can bind to a receptor that has what is called the death domain inside. And that can cause this second messenger signaling to happen. Apoptosis is activated that then finishes on downstream here. There are a number of factors that can be secreted during your embryonic development that causes cells to undergo apoptosis as a normal function of human development. Because there was a point in your developmental past that your hands and your feet looked like fins and flippers and paddles. The tissue between where your fingers and toes were going to form had to go away. It had to undergo apoptosis. Your esophagus was once a solid column of tissue. The cells in the middle had to undergo apoptosis and open up so you could swallow and breathe. So just a few examples of how this regulation and instructions of cells to undergo apoptosis is critical for your homeostasis right now as part of your immune system, but also during your embryonic development. What's the consequence of all that? It's like scissors or a weed eater coming into your DNA. It just shreds the DNA. Without your DNA, your cell's not going to do anything. It's not going to divide, but it's not going to be able to produce the new proteins that are going to be responsible for its continued survival. And I think you'd agree that would be a bad day.